The sperm swim over to the egg. The egg is surrounded by a barrier of glycoproteins called zona pellucida. The binding of the sperm to the zona releases enzymes that eat away at the zona, allowing the sperm to dig closer to the egg. When the sperm binds to the egg, it sets off a cortical reaction where cortical granules inside the cell release enzymes that turn the zona layer into a fertilization membrane, which prevents other sperm from binding. The membrane of the sperm binds to that of the egg, allowing the sperm to empty its genetic material into the egg. Note that the genetic material does not actually leave the cell, it was just removed for convenience.
The next step in embryonic development is cleavage. In mice, they go through spiral cleavage, meaning the cells/blastomeres form around the axis. The cells divide on the longitudinal plane, horizontally and vertically, until there are 64 cells.
The mass of cells then begins to compact to form a morula. Then they differentiate, forming an outer layer called trophoblasts in blue around the embryoblasts in orange. The inner cells cluster within the trophoblast ring, forming an empty space called the blastocele. The whole unit is called a blastula, and this is when gastrulation begins. The cells differentiate further, forming the teal primitive endoderm, the orange epiblast, the blue trophectoderm, and the green distal VE. It is around this point that the blastula hatches from the zona pellucida. The blastula then begins to fold in on itself, forming a gastrula. It then further differentiates into a pink layer called the anterior visceral endoderm and a tan layer called the primitive streak.